Okay, let's start.
Good
So,
thing it's at night.
for those of you who didn't hear when I was talking to some of the students here at the front of the class a few minutes ago, my plan is to get through chapter 10 before our exam on the 3rd. So that's the rest of this chapter in meiosis, which hopefully we'll get through most of today, which will leave us, when we get back, about a week to get through genetics. And I think that that should be sufficient for that. And then after that exam, there'll just be another couple of chapters that we'll go over before the end of the term. A couple of you have asked about the possibility of quizzes. So some of you have prepared for a quiz last week, and then I totally dropped the ball and forgot to post a quiz. So I will post a quiz for this week, which is terrible, I know, because it's Thanksgiving week. But I'm going to make the quiz available from Thursday through the end of Sunday. So you'll just have any time over the weekend to take it. And then to make up for the quiz that we lost this last week, I'm going to put a bonus quiz available for both the face-to-face and the online sections. It'll be bonus, which will mean that if you get items wrong, they won't count against you, but if you get them right, they will count for you. Yeah, so there'll be a possibility of 20 points extra credit there.
So will that be on 7 and 8 in Elephant
I
Science?
don't know.
Or
I mean,
just
we
7?
can. I won't post that this week,
Okay.
and we can make it out over Elephants on Acid if you want, or we can make it over just lecture content, or we can kind of design it how you guys would like to see it.
What chapter will this week be over?
So it'll be over chapter 7, the one that we didn't do last week.
What chapter in the
7,
Oh, okay.
the one that we didn't do last week. Yeah.
Okay.
Other questions?
They're not both extra credit, right? Just
No, just one will be extra credit. The one for this week will be just a normal quiz. Other questions? All right. So last time we were talking about mitosis, and we concluded with this slide right here. There is a process that cells go through when they've been damaged or something isn't going quite right, called apoptosis, and it's programmed cell death. And this is important for a couple of reasons. The first of which, right, is embryonic development. That was kind of the context in which we talked about apoptosis. So there are structures in our embryonic development that we have there that we don't have as adults, like a post-anal tail and webbed fingers and toes and things like that, which are kind of relics of evolution past. So when we're getting rid of those, what our cells do is they initiate a whole biochemical pathway in which cells that constitute those structures, like the webbing between digits, kind of gets digested. And then all of those cellular products are recycled into other sorts of structures that we're developing at the time. This is also really, really important for cancer inhibition. So all of us at some point in our life will have a cancerous cell. But our bodies are really, really good, for the most part, at identifying those. And if we can repair that cell so that it's no longer cancerous, then that's what we'll do. If we can't, then we're going to cause that cell to go through apoptosis and just kill it off. Okay? So before we talk about cancer proper, what we need to do is talk a little bit about how the cell cycle is regulated. And it's a series of checkpoints, really, that we use. Now, it's important for us to understand at the outset of this part of the discussion that the default position for any cell is to not divide. So, for example, once a cell has gone through interphase, then mitosis, then cytokinesis, it receives a stop signal, and it just doesn't progress any farther. And that's the default position, really, for all cells. Really, in reality, they'll go through the G1 phase of interphase, but they won't complete interphase. And that's called arrested development. So the default position is to stop and to not divide. What then has to happen is something called a growth factor needs to be received, and the cell cycle will continue. But that growth factor is really just overcome the default position and then progress. So there are 3 main checkpoints in the cell cycle, and the first of which is the G1 phase. At the G1 phase, at the conclusion of the G1 phase, there is a stop signal. And then what happens is some regulatory proteins survey the landscape of the cell. And if they find that everything is going well, all of the proteins that are supposed to be replicated are being replicated, and the organelles are being replicated, and the DNA at this point looks okay, then a growth factor will initiate the next phase. And you'll pass through G1 and go into the S phase of interphase. And then there is a checkpoint at the G2 phase. The G2 phase checkpoint is an important one, though arguably the most important of them is the G1 phase. Generally speaking, if a cell gets past the G1 phase checkpoint, it will complete the cell cycle and go all the way through cytokinesis. So a less important one is the G2 phase. And at the G2 phase, this checkpoint, really what's being assessed is whether or not the cell has a complete complement of organelles that will facilitate like sustained life in two identical daughter cells, but more importantly, that the DNA has been replicated correctly during the S phase. If the DNA is not replicated correctly during the S phase, then another sort of subsystem is initiated. And in that system, the DNA is repaired, or at least the cell attempts to repair it, and if it can, then it will pass through the G2 phase checkpoint. If it can't, then apoptosis and the cell will be killed off. And then the final checkpoint is in mitosis, and that's called the M checkpoint. And this actually happens at metaphase. Remind me what occurs at metaphase in mitosis. Do you remember what that phase looks like? Yeah, everything's lined up in the middle of the cell, right? Those chromosomes are attached to each centriole, and they're all lined up at the center of the cell. And if you'll remember from the lab that we did last week, that's really the longest phase, right? And that's the longest phase because this is where an important checkpoint is initiated. If everything is lined up correctly, then the cell passes through that checkpoint and then completes cell division. And then we'll go through G1 and enter the arrested development stage again. Why is it, do you think, that M checkpoint is such an important one? What do you think, Carly?
Last time we talked about how if one of the chromosomes is too far to the left, too far to the right from the centriole, it causes them to break apart asymmetrically, if I can use two words, so that you have an extra chromosome on one or left.
Okay. Yeah, it's called a non-disjunction event. So ideally, what's going to happen, right, is we're going to have all of our chromosomes, and I'll only draw 4, lined up at the center of the cell. And if they are lined up at the center of the cell, what that means is the mitotic spindle that has centrioles on both sides have connected to all of the chromosomes. Now that's important for the next phase, anaphase, right? Because in anaphase, what happens is the cohesins that are holding those sister chromatids together are cleaved. They're broken apart. And then sister chromatids segregate to opposite ends of the cell. If things aren't lined up correctly, if, for example, one of these chromosomes is over here, really what that means is the mitotic spindle is not complete, right? This microtubule hasn't attached to that one yet. And if it hasn't, that means at the stage of anaphase, you're not going to get those two sister chromatids pulled apart, which means that this cell is going to end up with 3 chromosomes, and this cell is going to end up with 5 chromosomes. It's going to have an extra copy of that one. And non-disjunction events are crazy scary. I mean, that's how you get like Down syndrome and other really bad things.
Did you say that they fight until it gets to the center?
Yeah, and so what's supposed to happen, right, is as soon as this attaches, it's going to pull it towards the centriole, and then this microtubule has to grow out and connect to it, and then they enter like this tug-of-war thing. And the tug-of-war should always end in a draw with the chromosomes at the center of the cell. Now I should also mention, again, that metaphase is important because this represents the completion of the mitotic spindle. The mitotic spindle is not complete until metaphase. And so really, That's what that checkpoint is checking for, whether or not that mitotic spindle is indeed complete. Okay, so just a very brief recap. Remember, the cell's default position is no division. And then once each of these checkpoints are reached, something called a growth factor, which we'll talk about on the next slide, I believe, will be initiated. And then that checkpoint will be overridden, and the cell will be able to continue into the next phases. Okay? All right. Oh, let me give you one more analogy for checkpoints. Do you guys know? Do you guys know how washing machines work? So washing machines, right? You throw your clothes in them and then you put the soap in and they wash your clothes. But they go through distinct phases, right? I mean, there's a fill phase, then there's an agitation phase and a spin phase, and then depending on how clean you want your clothes, there's another fill, agitation, spin, right? Rinse and repeat type stuff. So there are actually two checkpoints in washing machines. The first one is just a duration of time. A certain amount of time has to elapse before the next phase can be initiated. So your washing machine knows how long it should take to fill that canister, that barrel that it's filling. But there is a secondary check system, right, where they're actually making sure that the water level has reached a certain level. And that's exactly the same thing with the cell cycle. The cells know about how long they should be in each phase. And so these checkpoints and the growth factors that we'll talk about next won't be initiated until a certain amount of time has elapsed. Once that time has elapsed, there is an arrested development phase, and then these survey proteins are released and they check and make sure everything is okay. So it's really a two-system checkpoint, right? There's an elapsed time, and then there's a survey, just like our washing machines. There has to be a certain amount of time spent in filling and in agitating and in spinning. And then once that time has elapsed, then the washing machine can actually do its job and look to see whether or not the water's at the right level or the water has been drained out or whatever. Okay, so let's talk about growth factors just really quickly. A growth factor is really just a protein. It's a chemical signal that's released by an adjacent cell that tells that adjacent cell to divide. Now, growth factors bind to surface proteins on the outside of the cell. So really, that signal to divide is coming from outside the cell, at least that initial signal. Now that can happen when a cell is perhaps damaged or during distinct phases of our own physical development, right? So like during puberty, for example, you have lots of growth factors that are being released that are stimulating growth. If we get cut, then what happens is the cells just adjacent to the damaged cells will start releasing growth factors, and then those cells will begin to divide and repair the wounds that we've incurred. And really the job of a growth factor is to override that stop signal at the checkpoint. Okay, you okay with that idea? It's just a protein. Protein is released by another cell, and then that protein interacts with the surface of the cell that needs to divide. It receives that signal, and then it will start going through the cell cycle. Now there is some specificity to growth factors. They can either be very general, which is commonly the case with pubescent development, or they can be incredibly specific. And it all depends on the shape of the protein and the type of receptor that that protein is interacting with on the cell. So let's think about this contextually, right? We have a cell cycle, and there are distinct regulatory stages. There are checkpoints that need to be passed. The system is in place so the cell cycle doesn't just go crazy, and we have uncontrolled cell growth. We have these distinct phases, and at each distinct phase there is a stop signal. You can't go beyond this point until some growth factors are released to make sure that the cell is healthy and that it can pass those distinct checkpoints. So this is our regulatory system. That G sub 0 phase, or the phase of arrested development, will vary considerably in time depending on the type of cell that we're considering. So there are some tissues that have a very, very long arrested development phase and a very, very short arrested development phase. You guys have probably heard, right, that brain damage is irreversible because neurons can't be replicated, and that's not true. Neurons do replicate, but they replicate incredibly slowly. So their G sub 0 phase is incredibly long, which is why dementia is such a scary thing. That's a neurological disorder where you're actually losing neurons in the brain. And your body can't regenerate them as quickly as you're losing them because of how long that G sub 0 phase is, that arrested development phase is. But there are other tissues that we have that have a very, very short arrested development, or a very, very short G sub 0 phase. And they're generally tissues that are abraded with some frequency, like our skin, right? We're constantly rubbing our skin up against things and it's sloughing cells off. Consequently, our epidermal tissues have a very, very short G sub 0 phase, and we're replicating skin cells frequently. Same thing with our GI tract, right? We're constantly swallowing things, and it's scraping cells off the surface of our GI tract. Because of that, we have to replicate cells in the GI tract with some frequency. In fact, it's estimated that you replicate like your entire GI tract every other day or so. So very, very short. Okay, questions so far? So this is as much as I wanted to talk about the checkpoints and the regulation of the cell cycle. We're going to start talking about cancer in just a second. But on an exam, you ought to expect questions like, during the G1 phase, which of the following things happen, right? And at which of the following checkpoints do we assess whether or not chromosomes are aligned correctly? Or at which checkpoint do we assess whether or not the DNA has been replicated correctly? So